Hello, my name is Jared Hardner. This presentation is about conducting biodiversity baseline studies that will be compliant with the 2019 update of Performance Requirement 6 of the European Bank for Reconstruction and Development, or PR6 for short. This presentation is part of a series that walks the viewer through all stages of developing an environmental impact study and mitigation plan that will comply with PR6. Viewers should also consult the 2020 guidance note for PR6, available on the web. A key concept for designing a good biodiversity baseline study is iteration. Most biodiversity baseline studies are composed of 4 general stages: defining a study area, scoping the study, conducting field work, and identifying areas that require special attention in impact assessment and mitigation planning. In the case of PR6, we call this last step critical habitat assessment. I will explain each of these steps in more detail, but before I do, I want to emphasize that these steps should not be conducted in a rigid sequence. The study should be designed to allow the analyst to return to any stage of the baseline study based on what they find as they work. For example, field work can generate unexpected information about important species present in the area of the project's influence. It may be necessary to return to the first stage and redefine the study area whenever this happens, so that the landscape distribution of these species is adequately documented. Designing for iterative study means that the clients for the baseline study must understand that timelines and budgets should not be fixed at the outset of the study. They must be flexible. Let's go on now to discuss each of these stages of the baseline study. The first stage of the baseline study is to define the study area. As I mentioned a moment ago, the study area will be defined iteratively as information is gained about the site. For the first pass, the study area can be defined in terms of the area of the project's influence. This is typically determined by another part of the team performing impact assessment for the project, rather than biologists conducting the baseline study. It will generally include the physical footprint of the project, shown here in black. The direct footprint is extended to consider other direct impacts, such as emissions, effluents, dust, noise, and light impacts, as shown in red. If a project induces other impacts, such as other development near the project, this must be included as well, as shown in orange. Now draw a polygon that encompasses these impacts and follows sensible ecological boundaries, such as a watershed boundary. This is the boundary of your core study area. Remember, this is going to be refined in the subsequent stages of the study. Before we proceed to the next stage, I want to talk about a technique for defining a project's area of influence that is prohibited. Some people call it slicing. It involves conducting separate impact assessments for parts of a larger project. By slicing a project into smaller pieces, it is possible to conclude that none of the individual pieces have significant impacts, while in truth, if the entire project were considered, significant impacts would be apparent. In this example, it is obvious that it would make little sense to evaluate just a single portion of a highway improvement, since it is part of a larger highway. For PR6, this is true even if the EBRD is only funding one component of the project. This should be kept in mind when defining the study area. Scoping provides the basis for selecting the appropriate baseline study methods. It should identify the ecosystems, important biodiversity features, and ecosystem services in the area that may require focused study. Scoping should include an initial desktop analysis and literature review, including a review of regional studies and assessments and global or regional screening tools such as the Integrated Biodiversity Assessment Tool, or IBAT. Also important is a review of stakeholders who may be beneficiaries of ecosystem services generated in the area. This should be followed by initial field reconnaissance and consultation with relevant stakeholders and specialists. The types of features that should be identified for potential focused study include ecosystems that are priority for conservation, regularly occurring species and their habitats that are a priority for conservation, protected areas, and priority ecosystem services. The definitions and further details for each are included in the PR6 guidance note. We'll discuss a few of them in the examples that follow. But before we go on, it is important to note that areas designated as UNESCO Natural World Heritage Sites, or that meet the designation criteria of the Alliance for Zero Extinction, should be flagged at this stage. Projects that will adversely affect these areas will not be financed by EBRD, with the possible exception of projects necessary for the conservation management of the sites. Upon completing the scoping study, the study area may require refinement. For example, the study area may need to be expanded to fully encompass the ecologically appropriate areas of analysis, or EAAA, for features that require additional focus study. In most cases, that will mean the landscape level distribution of the feature requiring study, considering the ecological patterns, processes, and functions that are necessary to maintain it. If the feature has a patchy distribution in the landscape, it may be necessary to devise an area of analysis that encompasses multiple disjunct sites. For some wide-ranging species, the area of analysis may be informed by areas of aggregation, recruitment, or other specific habitat features of importance to the species. In all cases, the area should consider the distribution and connectivity of such features in the landscape or seascape and the ecological processes that support them. Where it can be shown that multiple values have largely overlapping ecological requirements and distributions, a common or aggregated EAAA may be appropriate. At this stage, EAAAs will be a first approximation based on the information available. They will be further developed and refined once fieldwork is undertaken. Remember, an EAAA follows the natural distribution of a species or ecosystem with ecologically sensible boundaries. It is never defined by a fixed perimeter around a project. An EAAA is often confused with the area of influence, or AOI, but they are different things. An EAAA tells us where things live. An AOI tells us where they might be affected. The AOI will become important during impact assessment, but for purposes of baseline study, the EAAA is the key unit of analysis. Fieldwork is necessary to complete the baseline study. Field surveys should characterize the general ecological context, including a map of aquatic ecosystems and terrestrial vegetation types, each with representative sampling of their species composition, structure, and condition in the core study area. Specialists in the relevant fields should conduct the work, following good international practice. For example, in this photo, ornithologists were needed to record occurrences of a threatened bird in patches of remnant forest habitat. The environment and social assessment teams should work in an integrated manner to collect information from relevant stakeholders on local ecosystem services and the degree to which they are relied upon, using appropriate methods for the social context. It is important to clarify that fieldwork should be commensurate with and proportional to the potential risks and impacts of the planned development. This means that not all things need to be studied in the same level of detail. Some types of biodiversity features, such as threatened species, deserve closer study. Use the map of aquatic ecosystems and terrestrial vegetation types to organize representative surveys of plants and animals. Some points to remember are: 1. It is important to record the GPS coordinates of samples. 2. It is also important to record when the sampling occurred and to ensure that sampling is undertaken in more than one season. This may mean the baseline study requires many months to be completed. 3. Recorded species should be organized according to the aquatic ecosystem or terrestrial vegetation type where they were observed. 4. The conservation status at the global and national level should be included. And 5. An analysis of the sufficiency of the sampling should be conducted, as shown on the right with the species area curve. As shown here, each vegetation type is sampled until the cumulative species curve begins to flatten. If fieldwork reveals additional information about the biodiversity features in the area, refined EAAAs may need to be developed. In some cases, new areas of analysis may be added. In this example, fieldwork revealed the presence of a previously unexpected threatened plant species. This species will need to be studied more closely, requiring its own EAAA to be added to the study area design, as shown here. Once the areas of analysis are determined for all species requiring additional focus study, specialists can then design and conduct such studies to inform the impact assessment with relevant information such as the relative abundance of a threatened species and its use of the habitat in its respective area of analysis. The information should also be sufficient to inform mitigation planning. This will include the features of greatest importance, such as breeding areas, to inform project layout and to avoid or minimize impacts in these areas. With the information collected in the baseline study, you can proceed to the next stage, critical habitat assessment. The purpose of this stage is to determine if any features in the study area qualify as priority biodiversity features or critical habitat, following the definitions provided in PR6 and Guidance Note 6. These features will require special attention in impact assessment and mitigation planning. But please note, this is an assessment of the context in which the development is proposed, and therefore does not consider specific impacts at this stage of analysis. It answers the basic question: how important is each EAAA for conservation, and what PR6 requirements will apply? To perform this assessment, the list of all features identified for focus study and confirmed to be in the study area should be tested against the criteria in Table 1 of Guidance Note 6, as partially shown in this slide and continued in the next slide. The criteria and conditions detailed in Table 1 are based on the EU Habitats and Birds Directives, the Berne Convention, and/or drawn from IUCN's Key Biodiversity Area Standard. This is a continuation of the table showing species-level criteria. Some criteria have no predetermined conditions, i.e., PR6 paragraphs 12.3, quote, significant biodiversity features identified by a broad set of stakeholders or governments, and paragraph 14.5, quote, areas associated with key evolutionary processes. For these criteria, the assessment must rely upon expert judgment beyond the use of this table. Ecosystem services are not explicitly included in this table, but should be prioritized in coordination with the social baseline analysis, according to the importance to and dependence on the ecosystem services by the relevant stakeholders. So, here are some examples of critical habitat assessment. The large mammal satisfies none of the conditions in Table 1 for critical habitat. But because it is listed in Annex 2 of the Habitats Directive, it is designated a priority biodiversity feature. The rare plant is range restricted, and the EAAA holds greater than 10% of that species' global population. We determine this by using area as a proxy for population, where the area of the EAAA is divided by the total area of occupancy. If area of occupancy is not known, the extent of occurrence should be used. There are no quantitative thresholds for species that appear on a national red list but are otherwise not on the IUCN's global red list or on one of the annexes of the Habitats Directive, Birds Directive, or Berne Convention. So it will require the expert judgment of a specialist to ascertain if the local population is of significant importance for the persistence of that species at a national level. If the answer is yes, the EAAA should be designated as critical habitat. That's the case with the amphibian in this example. Lastly, the disjunct habitat identified during scoping turns out not to be regularly occupied by the species of concern, so neither a priority biodiversity feature nor critical habitat designation is necessary. It merits mention that additional baseline study may be necessary to refine this assessment, so the baseline team should be prepared to do so, and the client should be aware of the potential cost and time required to complete the critical habitat assessment. Following is a list of the critical elements of the baseline study. Once completed, it is very helpful to share the data with the scientific community to inform the overall understanding of biodiversity in a region. If all projects did this, it would eventually lower the cost of all biodiversity baseline studies, something for a project developer to consider. Lastly, it is all too common for the raw data from baseline studies to get lost. It is not uncommon to need to return to these data in the future. find a way to store them safely, record their location, and back it up. For more information on biodiversity baseline methods, see Good Practices for the Collection of Biodiversity Baseline Data, developed by the Multilateral Financial Institutions Biodiversity Working Group, and available on EBRD's website. You can also visit us on the web at www.hg-llc.com. Thanks, and see you in the field.